aware nanoparticle for disease management is a budding science, and we all are eagerly waiting to hear Dr. Wade, who is a well-known expert in this area. I'll not take much time, therefore I'll request Sushil to formally introduce Dr. Wade. Before that, I'll congratulate IPS and Dr. Sharma for taking this initiative and giving me opportunity to welcome Dr. Wade. Sushil, you can start.
Thank you very much, sir. So I have a privilege to welcome Dr. Wade Elmer, who is the very distinguished speaker of the day on this IPS Platinum Jubilee lecture series. To briefly highlight his achievements and his brief bio sketch, he is working at Connecticut Agricultural Experimental Station as Vice Director of the station and also Chief Scientist of Department of Plant Pathology and Ecology. He has received his PhD from Michigan State University in the year 1985 and started his career from the same experimental station in Connecticut in the year 1987. His main area of work mainly focuses with a goal to develop the strategies for disease suppression of ornamentals, vegetables, and field crops using combination of minerals, nutrients, soil amendments, earthworms, and biocontrol agents. In the past 8 years, he has been able to develop advanced strategies of using metal oxide nanoparticles to improve the overall plant health. Dr. Elmer has edited 4 books, authored and co-authored over 120 peer-reviewed publications in very high impact factor journals, 20 book chapters, and has written more than 100 articles in different magazines and symposia. He has to his credit a number of prestigious awards, including Award of Merit by American Phytopathological Society. He is also a fellow of American Phytopathological Society, and he has been awarded Outstanding Paper of the Year Award for 2012, and he is also a visiting scientist at Royal Botanical Gardens, Australia, and University of Sydney, Australia. So in addition to many achievements, I have a privilege to welcome him and also invite him for today's talk.
Well, thank you very much. It's a real honor to be able to celebrate this momentous occasion with the Indian Phytopathology Society. I want to congratulate you on 52 years. Is that right? In this endeavor. So I'll go ahead and begin. Share my screen, and hopefully everybody can see that. Can you see that? Not yet, probably.
Yes, it's visible.
You could, okay. Let me put it into slide mode.
Yeah.
There. Okay. Yeah, well, thank you. The topic is it, can you see everybody can see it okay?
It's okay, fine.
Okay. Excellent. Okay, the topic I'd like to talk to you about is something I've been working on quite intensely for about the last 8 years, and I kind of stumbled onto it, and it's been a really exciting area. I really wish I was 20, 30 years younger so I could just spend the rest of my career developing this further. But it's about using nanoparticles of copper in plant disease management. And as I was mentioned, I am the Vice Director and Chief Scientist in the Department of Plant Pathology in Connecticut. I've been here for 34 years, and it's been a really wonderful career that I've had. I want to just first mention things you're probably already aware of, but you know the human population is going to be increasing quite high by the year 2050. The estimates will go from quite a range, but it's guaranteed it's supposed to about double. And we feel our food production is going to need to be able to increase significantly. And this is going to affect many countries like India as much. the increased food, they say, is more likely is going to come from intensifying current acreage than through area expansion. And that's based on a lot of published reports. The climate change models are also predicting some real drought and heat stress is going to have a major effect upon many of our grain estimates. So I think it's fair to say, and I think you all know this in plant pathology, that we have some very daunting problems ahead of us in plant pathology. On top of that, we now know that pesticide and nutrient efficiency is very poor, and we need to improve upon that. So the big question, as you know, is how do we increase the production in these areas without adversely affecting the environmental outcomes? This has been a real challenge for us. And one of the things that I took on is in my career early on was looking at how mineral nutrition could affect plant diseases. And I began working on this during the 1990s and such. And then in 2007, I and some colleagues put together a book that was published by the American Phytopathological Society called Mineral Nutrition and Plant Disease. And in this, we found experts to talk about many of the nutrients that plants need in order to complete their life cycle, and a few beneficial elements, such as like aluminum and silicon, things that were not much was known about them back then. And one thing that became very surprising to me as I was writing and reviewing many of these chapters was the role of micronutrients. It seemed like whenever a system, be it mineral nutrition or biocontrol or whatever it is, whenever a system improved plant health, it had a direct effect upon the availability of micronutrients in the roots of these plants. And I became very intrigued with that. So I started looking at micronutrients. As you know, they are very important in protecting roots against soil-borne diseases. They activate many enzymes that create defense products. The second a root-infecting organism invades those cells, the root usually responds immediately by laying down defense mechanisms, lignins and so forth. And many of these enzymes that create these products are activated by micronutrients. Copper, as I'll talk about most of this talk, it really activates the polyphenol oxidase pathways, along with many other things. Manganese is known to activate enzymes in the shikimic acid pathway and the phenylpropanoid pathways. And these are the first committed steps, in creating lignin. Zinc is, as you know, is very important in activating superoxide dismutases, which go around and protect plants from the free radicals that form through stress, through drought and saline stress. So these micronutrients are very important in the overall health of a plant. One thing about micronutrients, as you know, if you study these, is that they are not precipitally translocated. So if they're sprayed onto the leaves, they really do not travel down to the roots very effectively. So that's always been a problem. How do we get them to the roots? And this is where I really wanted to make sure I could get these elements there, because that's where the enzymes need to be activated to provide these defense barriers. On top of that, we found that when we apply these micronutrients to soils, at least most of the soils that we deal with here in the United States and perhaps India too, they're in the pH of about 5.5 to 7. There are many areas in here where we get above that, but at that pH, these micronutrients are not available. They precipitate out as oxides. And we find that we have to put on so much into the soil in order to make them available. It becomes environmentally insensitive. Also, as you look at the pH, this chart I have here shows the two red lines. This is the range in which we're growing plants, most of the plants we grow, and the micronutrients, the availability. This shows the availability as the pH goes from acid to alkaline, shows that the availability of these micronutrients really drop off here. So this has always been an issue. There usually seems to be enough micronutrients to prevent foliar deficiencies from being apparent, but we do seem to have a deficiency when they're under attack by a plant pathogen. So this is where we felt like we needed to improve micronutrient availability in the roots. And most of my career was looking at how cover crops, microbes, biochar, earthworms. Have done a lot of work with all of these different systems to see how they could enhance the availability of micronutrients. And I didn't get a whole lot of progress in any of those systems. But what I found with nanoparticles opened up a whole new avenue for me for trying to increase the availability of these products in the roots. This was a study that was done by a colleague in my station, Dr. Jason White, who I've been working with very intensely for about the last 8 years. He found that when the nanoparticles of copper oxide were applied, they to one side in a split root system, the nanoparticles went up, you know, as we would know, but they also were detected in the non-treated side. So there was systemic movement up into the xylem and then down through the phloem. Now, this was something I didn't think was possible, but apparently the nanoparticle form was such that it could actually travel in the xylem and the phloem, according to this one study. So this came out in 2012, and I immediately asked Dr. White, could we spray these on the leaves and get downward movement of these products to the roots? And of course, he didn't know because we've never done that. And more importantly, could we, if it did move downward, would there be enough effect upon a soil-borne disease that we could get suppression? So that was the big hypothesis that I had back in 2012. I began to wonder, how can nanotechnology begin to help affect plant diseases? Now, this is your classic disease triangle. As you know, you need a susceptible plant, a virulent pathogen, and an environment that's conducive in order to get disease. And I found that nanotechnology really had a direct effect upon affecting two areas. And that was the susceptibility of the plant and the pathogen. Many of these nanoparticles, like of copper and sulfur, and actually silver, although that's not used too much in the field, but these things also had antimicrobial properties. But I was more interested in how these micronutrients, nanoparticles of micronutrients, would affect the susceptibility of the host. And this is the area that I'm going to be looking at, is in this part of the disease triangle. I think as we learn more and more, we might find that nanoparticles actually can be part of making the environment more or less conducive to disease too. But right now, the studies have been focusing on the susceptibility of the host and as a bactericide or fungicide. Now, there are many types of nanoparticles that can deliver copper. As I mentioned, that's going to be the topic of my talk. And these are, in fact, I could update this. Today, I heard a seminar about a new type of nanoparticle called a mycene that is being developed to be loaded with copper as well. But most of the nanoparticles I work with are these small particles of copper oxide. We can also make copper nanosheets, and these can be made from copper oxide or copper phosphate. And they actually are small little sheets that lay actually flat on the surface of the leaf tissue, and they can dissolve copper faster than the oxides. Carbon nanotubes are another way that's been used. These are actually graphene sheets, very thin single-cell graphene single molecule sheets that have been rolled up, and they can be functionalized with copper along the outside. And this can be like a delivery system, and they can be single or double walled. So that's one thing. And then carbon dots is a new area that's been examined. These are actually small carbon nitrogen. The composition can vary considerably, but they're small, uniform size, and these things can also be loaded with copper and be used to deliver that into a system. Graphene oxides are just the single sheets that are used in making the nanotubes, and these things can be loaded with many things, including copper and such. Also, mesoporous silica. This is silica oxide nanoparticles that are being prepared in these small little, almost like little wiffle balls, and they are tremendous surface area in there. So not only do they release a lot of silicon, but they can be loaded with copper. So these are the various systems that are available right now for delivering copper. But I kind of diverged a little bit from what I was going to start showing. What I want to talk about more today is just 3 systems that I've worked on where I've been looking at the effects of copper. Of course, verticillium wilt of eggplant, that's a real major root disease problem here in Connecticut. I think in your neck of the woods, it's probably more fusarium on the bringle that you have here. But this is a major issue here. again, very similar to fusarium, causes a wilt and the drop of the lower leaves. Fusarium wilt of watermelon is becoming a bigger problem because growers want to grow these small, delicious, seedless watermelons. And they're very popular, they're very marketable, but they're also very susceptible to fusarium. So we're having to deal with a whole new problem that we pretty much had eliminated with some resistance, but we lost that when they decided to use these varieties. And I wanted to include one ornamental because very little research is being done right now with nanotechnology in ornamental research. And this is an area where a tremendous amount of fertilizers and chemicals are used in order to produce these perfect flowers and perfect plants for consumers. And it's a really rich area where I think nanotechnology could use to reduce a lot of the toxic chemicals we're putting into our environment. So beginning with verticillium wilt of eggplant, this particular study was done with using very small seedlings. And see, this is one of my coworkers here, and these are very small seedlings of eggplants. And these were sprayed with a water solution of these nanoparticles, about 500 parts per million. It's fairly concentrated there, but we're only putting on a few mils. So only about 1 or 2 milligrams of the nanoparticles are going on each plant. And these plants are then sprayed once, and that's very key. I'll probably say that many times during this presentation. These things are sprayed once as seedlings, then they're taken out, they're putting in the field. This particular field is very infested with verticillium, so I'm very sure I'm going to be getting disease. And then right around the time that they start to flower, and I'll talk a little bit more about that later on, they start showing the symptoms of verticillium. They start crashing then. But we do get considerable yield, and we collect that yield. And what I have down here is a graph just showing the yield of these plants. Now, in this study, we looked at nanoparticles of copper, manganese, and zinc oxide, but we also included the larger bulk equivalent. These are exactly the same product. It's just the larger form that you would get through Sigma or the standard supply houses and such. And what I want to show, what it shows here is the effect of the small size. What we found in 2013 and 2014 is that the plants that received copper oxide nanoparticles had more yield than the controlled, and they had more yield than the bulk form, the larger bulk form, and much more than manganese. And 1 year we saw it with zinc. Zinc was always very variable, and I have to admit sometimes we saw some effects with manganese, but consistently we see it with copper, and always the biggest effect. Personally, I found this so hard to believe that treating a very small seedling, putting it in the field, months later seeing an increase in yield. And it was really hard for me to understand how that could possibly happen. We published this, but I really wanted to keep doing the studies, but my colleague really wanted to get this out. So we went ahead and published that, but I wanted to keep working with the system. One thing we did is we took the fruit and we analyzed it to see if there was any differences in the elemental composition. In the upper right here, this was the control showing the distribution of those micronutrients. When copper was used, there's really no major differences in how these things were distributed, or with manganese or with zinc. So statistically, there was no effect upon the overall composition of the elements. One thing we don't know is whether this copper is in the nanoparticle form, or if the zinc or the manganese is in the nanoparticle form, or the zinc there. Most likely it's in the ionic form, but again, we don't have the evidence. It's really looking for a needle in the haystack when we look for these type of things. But I was curious, as I mentioned before, 2 years we saw the statistical increase with plants that were treated with the nanoparticles of copper when they were sprayed once as seedlings. And here's a picture of the field. Again, this plot right here that's circled is the nanoparticles of copper. We were instantly seeing an increase in growth and vigor when these plants were treated. Now I became curious to know, well, I've always known that you want to have a balanced fertilizer regime. So what about combining these nanoparticles? What about combining copper with manganese, with zinc? Would we get any improved benefit over that? So I began a very long study for about 4 years to investigate that. And we did this in the greenhouse in the field. We were looking at how do these combinations affect growth, yield, and yield of eggplants when they were exposed to verticillium wilt. And we had the treatments. We had each nanoparticle, copper, manganese, and zinc by themselves. Then we had all the different combinations. We did this in the greenhouse and the field. In the field, we had it treated at 500 parts per million. But when we combined them, we had 500 plus 500. So there was actually 1,000 parts of copper and manganese put on these treatments here. And then when all three of them had it, was 1,500. Now we were criticized by having so much product there. So we did a series of greenhouse experiments where we reduced the rate so that the total amount equaled 500 ppm. And we ran all these together in the greenhouse several different times. And here we looked for fresh biomass, disease severity, fruit yield, and of course the elemental tissue composition. And in this is one of the first greenhouse studies I want to show here. The black bars are those treatments that received 500 ppm, and it was added together to make up 1,000, or in the combination of all three, maybe 1,500. And the gray bars are those treatments that the total amount equaled 500. But the point is, it really doesn't matter. What we found is that nanoparticles applied once by themselves at 500 ppm gave us the most reduction in disease. This is the area and disease progress curve. So the larger the number, the more disease. That's where we got the most reduction in disease symptoms. We also saw it when copper was combined with zinc, but there was no further increase. We got about the same amount. But what's so important is all the other combinations, there was no statistical difference from the control. When you looked at the biomass of the plant, again, only the nanoparticles of copper gave us plants that were larger than the control. And all the combinations at all the different concentrations had no effect. So that was very striking. So we began to think there might be some type of maybe a competitive exclusion going on here, where there was just a competition for sites on the leaf. And if you started adding in other nanoparticles, you might both be blocking that. So it seemed like just the copper by itself. This was some of the field work we did, again, with the same combinations. And again, we found that in 2016, 17, and 18, in 2 of those years, the nanoparticles of copper gave us a statistical increase. We didn't see it in 1 year. And 1 year we saw that with manganese, 1 year we saw it with zinc. But again, the take-home message is that combining them did not give us any more statistical increase versus the control. So it was basically the best treatment overall in all these 3 years was just copper by itself. If you go back to the greenhouse study, we took the leaves from these plants, and again, we treated these very young plants, and later on, 8 weeks later, we have a very large plant that we washed in distilled water, and then we analyzed the leaves to see how much copper there might be distributed in there. And we found that plants that only had nanoparticles of copper oxide had the most copper. We also saw it to a small degree when it was combined with zinc and with manganese at both the higher concentration and the lower concentration. But you remember, this was the best treatment, and it also had the most copper in it. So we kind of felt like any treatment that's going to put the most copper into that plant seems to be associated with the most disease suppression and the most yield. And when you start combining them with other nanoparticles, you tend to reduce the amount of overall copper in there, and that may be why they're not as effective. So what I've done here is I've taken the yield from all 5 years where I did this study, 2013, 14, 16, 17, and 18, and combined it there. As you can see, that 4 out of 5 years, plants that treated with nanoparticles of copper had yield that was statistically greater than the control. 2 of those years we saw it with manganese, 1 year with zinc. So it seems like, it all depends, but the more consistent effect has always been with copper each year. And this is a trend I see over and over again. And we calculated that a single application of nanoparticles, this cost in America, we buy our nanoparticles from a supply company. We found this costs less than $20 per hectare. It would cost less than $20 to treat enough young seedlings to plant a hectare, and that would generate up to $6,300 more profit per acre for a grower in New England. And I thought this was just astonishing, and I'm still trying to convince growers of the excitement of this. Mainly, and also one of the main reasons is the safety aspects of using nanoparticles. So that's one reason why I think it hasn't really caught on, and consumer acceptance about nanotechnology. Now, one question we ask is, why does the nanoparticles of copper affect the disease progress? What could be one of the mechanisms? Well, we found that the symptoms of verticillium wilt on eggplant would always show up when the plant started to flower. And this is about 5 weeks after planting, which is about the middle of July for us. We usually plant the first week in June, and then we start seeing these symptoms start to build up there. And this is, we think this is when the source-sink relationship between the roots and the flower shift, and the fungus, which is living in very low densities in the roots, all of a sudden begin to advance into the phloem or into the xylem and move up. Now, plant here, you can see this is an untreated plant at flowering. The FB stands for flower buds, and they're very small, but you can see them in there. But you can already see symptoms starting to show up. You got defoliation of the leaf and so forth. And then right next to it was a plant treated with the nanoparticles of copper. Again, no symptoms, but they are flowering. So there's something we think that was keeping the fungus from advancing into the tissue there. And right now we're doing a lot of proteomics and metabolomics to look at the sap to see if we can find any particular differences between these, to see if there's maybe an antimicrobial product that's being produced by the plant that's fueled by a copper enzyme, or some other mechanism. We don't know yet. But we've done this over 3 years now, and it's been really striking. I'll be writing this study up soon. And then again, and here's what we found in the data. This is the percent colonization of the stems by the fungus. What we did is that we sampled the bottom part of the stem and the top part of the plant before flowering. And before flowering, we found very little incidence of the fungus. We found a little bit in the control before, which was surprising that we see a tiny bit in there. We found none in the upper stems. But then after flowering, we started to see that the control plants already had about 38% of the stems were colonized by verticillium, whereas those plants that were treated with nanoparticles of copper had very, very little. And then the upper stems, there was less, but it does show that the fungus was already moving into these plants. But when they were treated with copper, there was none. So we do see that we're definitely stopping the ascent of the fungus in the stem tissue by there. But it is in the base, it is in the roots, but we're seeming to stop it from advancing in. So we do think there's some antimicrobial product being produced by the plant that's inhibiting it. But we've yet to identify exactly what that is. We've been looking for chlorogenic acid, fumaric acid, all the basic phenolics that we know are toxic. But so far, we haven't found any evidence for those yet. But we're going to keep looking. The next system I want to talk to you briefly about is, again, fusarium wilt of watermelon, caused by Fusarium oxysprum and typical macroconidial spores from here. This is a difficult disease to show differences in because frequently you'll get fruit set before the vines collapse. You can see here. So it's hard to see big differences in yield many times because you will get fruit. They'll be smaller and they may have other problems and so forth. But in terms of weight and so forth, it was a difficult system to work on. But I had a small grant to work on it, so that's why I chose it. Now, what we found with watermelon, as soon as we treated these young plants in the greenhouse with nanoparticles, again, we saw this growth burst. They started to grow. They started to run faster than the controls. And then once they were inoculated with fusarium, we found at the end of the system there, the ones treated with nanoparticles of copper were certainly doing much better than the control. We did a lot of research where we took the roots of these plants and did some transcriptomics. I'll show that later on. But the first studies that we did here, just to show again how that we're not just jumping in on copper, we originally looked at how copper, and again, we had the nanoparticle and the larger bulk form of copper, manganese, silicon, titanium. Now, titanium was involved in photosynthesis systems and such, but I was curious to know if it had any effect in the disease progress. And then zinc. And lo and behold, when you look at the disease progress, or the AUDPC, we found that the plants that got the nanoparticles of copper were the healthiest and looked the best in the study. And all the other macronutrients in the nanoparticle form or the bulk form were less effective. So this became to a study here where we treated young plants in the greenhouse. We also wanted to see what a second spray might do. So in this study, we came out here and we actually put out with a small mist sprayer, we actually treated them again after putting them into a field that was infested with Fusarium oxysporum, the pathogen that causes the wilt. And in this study, we looked at nanoparticles of boron. We included that, copper, manganese, and zinc. And again, what I'm going to present here is just on the yield. This was a study that we did, and we had plots that had no pathogen in it, and those were represented by the black bars. And you can see, regardless of the nanoparticle, there was no difference in the overall yield in terms of statistical significance. But when they were planted in plots that had Fusarium, lo and behold, we found plants that received the copper oxide had yield that was comparable to the controls. So the copper was able to completely overcome the damaging effects of the pathogen. So this was, again, real surprising, and this was just 2 sprays in this particular study. And again, if you want to look at the overall, the rank ratings of the disease here, we found that, again, the boron, the manganese, and the zinc did give us some suppression, but copper oxide gave us the most. And that's where we saw the most, the benefits in terms of yield. So once again, copper oxide tends to, rise out of the pack and to be the best treatment, But we keep looking at all the other ones as well. Once I was giving a talk to a grower, and they were saying, "Oh, so I can just put on a copper fungicide and get the same effect." And I was saying, well, I wanted to test that. So in this study, we looked at a control, the nanoparticles of copper, the larger bulk form. And then we used 2 copper fungicides, Coside 2000, which is a copper hydroxide, and then a copper octanoate, which is it's an organic form of copper and so forth. So then these were all put on the exact same amounts of copper to young plants, and then they were put out in a field that was infested with with Fusarium. And these plants were applied just once in the greenhouse with these various treatments. And this was done in 2015 and 2016. What we found in both years is that we had more yield in those plants that were treated with the nanoparticles of copper. I found this very hard to believe. Statistically, we didn't see any difference in either one year, but if you combine both years together, we were able to show a very slight statistical increase with nanoparticles of copper. And I found that just very striking, that just treating a very young watermelon plant in a greenhouse, planting it in a field with infested pathogens, I was able to see a yield difference later on. I think one of the reasons that this is having effect is that copper is an essential element that activates the polyphenol oxidase enzyme. The enzyme itself requires 4 copper molecules in the actual enzyme, and during the process of creating of this enzyme, it's able to lay down phenols and metallins and lignins and so forth. And these are the things you'll see, like when you bite into an apple, the browning that you see shortly after. Those are polyphenol oxidase enzymes creating that browning effect. So it's a very quick reaction that takes place. And we think that if we could get this enzyme in high levels early in the life of the plant, we might be able to delay that initial infections and such long enough to allow a plant to get a little bit bigger, have a little bit more vines, a little bit more growth, a little bit more flowers, and therefore more yield. At least that's what we think. There's a certain window there available early on in the young plant where copper might be able to enhance the health of the plant. And we showed this by doing some transcriptomics. Again, going back to these plants I showed earlier, when these roots were assayed for to see whether the genes that produce polyphenol oxidase and the PR1, the plant-resistant genes, are being increased. We found that when they were exposed to both the pathogen and copper, we got a significant uploading in both of these enzymes. So it did give us some support that we are definitely activating a defense mechanism for these plants early on. And we also did a similar study with tomato. And here we did a time course where we took samples before the onset of symptoms, and then we took them right after the onset. And in this study, we saw a significant increase in polyphenol oxidase in these tomato plants before symptoms even showed. And you can see here, this is the uploading is tremendously higher than we see when copper, and this was a copper phosphate nanosheet. It's a different form of copper. But again, in this particular situation, we saw that the copper and the fusarium together gave us a significant uploading there. So we do think we're turning these things on before the pathogen can start doing some damage, and that might be how we're able to protect the plant, again, for another period, a small period of time until the symptoms show up. Now, it always raises the question, well, why don't we go back and spray it again and every week? And we have done those experiments, but they don't seem to work as well. It seems like there's that one opportunity when the plant is young for this treatment, and after that, we don't see much benefit, which is kind of it's a shame that we couldn't be able to increase it better, but it does show that a very small amount applied to these very, very young plants early on, very little cost to the grower, very little threat to the environment, and we can get this benefit in the field. The last system I want to talk to you briefly about is, though in detail, is this nanoparticles on fusarium wilt pot mounds. And again, this is a major crop in Connecticut, as it is in India. It's a very important flower. And these cuttings I received, I rooted them, and then I sprayed them with copper, manganese, zinc. We used the standard fungicide, and with the and then the untreated. Now, none of these were inoculated yet, but you can already see the vigor once again with the copper. So there is really a growth enhancing effect that we're seeing on these plants early on. And this is striking because we've seen this in many different systems. In this study, was done in 2017, 18, and 2020. Again, looking at the disease ratings, the controls had the highest. We found the lowest disease ratings, once again, with the nanoparticles of copper. We see occasional benefit with some of the other ones, and of course with the fungicide as well, but copper always seems to be the best treatment that we come across. Here's showing some plants here. These are the non-infested controls. These were the young plants. They were starting to fill in, but haven't shown a lot of robust growth yet. When they were treated with nanoparticles of copper, already they're starting to looking much better. Manganese also had a good effect in this system. Zinc, and then the fungicides. Now, when you added the pathogen, here you can see the fungus is already starting to cause the necrosis and the chlorotic tissue in there. But the nanoparticles of copper tended to overcome that somewhat. Zinc and manganese were less so. The fungicide also gave us some control, but the nanoparticles of copper outperformed the fungicides in this study. And at the end of the study in 1 year, we have the controls, which none of those are particularly marketable. The ones treated with nanoparticles of copper, there are many marketable plants in there, even though the fungus is there at a high rate. Manganese and zinc had less horticultural value at the end, and also the fungicide had a few there. So not a lot of benefit to using the fungicides in this study. And this is something the growers here know. They know that if fusarium comes in, there's not a lot of good treatments that we can put on. It's really hard to arrest the disease once it forms there. But the nanoparticles of copper gave us some real promise for a potential mechanism for control. And again, looking at dry biomass in these studies for the 3 years, again, you can see that the untreated controls, the ones that were inoculated, the gray bars, and the controlled, the uninoculated ones, were always the smallest. And copper, in the first year, copper, manganese, and the fungicides gave us an increase. In the second year, we only saw it with copper, And then the third year, we saw it only with copper and the fungicide and only in the infested. So there's lots of variability. As you know, when you do field work, you're going to have a tremendous amount of variability. But this is why it's so important to do these studies over and over again. But regardless, copper, nanoparticles of copper tend to be pulled out of the pack, and they too seem to be the best treatment available to us right now. And this study we just got published this year. Now I want to show quickly a lot of other systems that I've worked on because these things are interesting. I've only worked on these, I've only done one experiment with these, so they haven't been repeated. But again, the same trend is showing here. This is Fusarium wilt of begonia. And again, this is a fungus called Fusarium foetens. And it's a new species that was shown up here. And I found it in Connecticut about but more like 15 years ago. Here's the control plants. When they were treated with copper, we got a slight increase. As I mentioned, there's always some benefit to the copper. Fusarium dropped the overall fresh weight. But in this situation, combining the fusarium with the copper, we had plants that were larger than the control. Very striking effect. Labiopsis black rot of violets is another disease that causes a lot of problems in bedding plants that we have here. We have a large bedding plant industry. And again, in here, I put on nanoparticles of copper at 250 ppm and 500. I used a lower rate because I thought that 500 might be toxic to the violet, but it really wasn't. I didn't see that. But the 250 gave us a better suppression. And the roots there, if you look at these roots very finely under the scope, there's lots of little blackened areas here that you don't see at the higher rates here. And also with the fresh weights, we saw at 250. So a completely different fungus, a different plant, but once again, the copper was beneficial. This is a study I've been doing in the field on pumpkin. This is the Halloween pumpkin that we grow here for our Halloween festivals. And again, in this study, I was combining nanoparticles of copper with mesoporous silica. As you know, pumpkins and cucurbits are active silicon accumulators, and it's also very beneficial for controlling a wide variety of diseases here. So again, I was looking to see if a combination would give us any improvement. What we found here, this is looking at this is the overall weight of the pumpkins coming off here. We found that the nanoparticles of copper and the silica, and when they were combined, were all beneficial. There was no interaction, in other words. We saw no additional effect by combining them. So once again, you could put on the silica or the nanoparticles and copper, and you could probably get the same effect. This particular field here, this is a control plant showing a complete collapsed plant, and this plant here was treated with copper. So you can see amazing effects there in the field. And we did this year as well too, but I just have not been able to put the data together. So our conclusions are nanoparticles of copper, they have growth promoting and disease suppressive properties that exceed their larger equivalents or the salts. And I should also mention we used a lot of chelated salts as well, and they did not work as well as the nanoparticles of copper. So it seems like it's the nanoparticle form is the superior form. Foliar applications to young plants require minimal product, and that reduces the exposure to soil. Since we're applying these to the foliage of young plants in the greenhouse, very little copper is being introduced into the soil or into the environment. And that has a lot of beneficial effects upon the environment. But the one thing that I think is the most important is that we're seeing season-long effects following a single application to a young transplant. I still find that astonishing, that we can treat this very young seedling and then months later see more yield in the field. And again, I believe that relates to that specific window when the plants are younger. And if we can protect them for that period of time, we can recognize, we can see the effects in terms of yield later on. The new future research that we're working on is looking for new tunable forms of nanoparticles of copper. You know, we're trying to design them. We've created ones that have a positive charge and a negative charge. Surprisingly, nanoparticles of copper that have a positive charge are ineffective. They don't have any effect at all, which is very interesting. So we're getting into the actual chemistry of the leaf surface and what's going on. Green synthesis is something that we are beginning to work on more now. because it's very important for making the availability of nanoparticles available to any other systems and so forth. We in the United States are working with a lot of chemists that are trying to advance the formulation for nanoparticles. And we're looking at various coatings that increase dispersion and dissolution and compatibility. We're also looking at a lot of biodegradable polymers. And these are very exciting because they can be put into the soil. As they degrade, they release the nutrient, and it could be anything from nitrogen to phosphorus, potassium, whatever, but it's a very slow release, and it can avoid the leaching of these products into groundwaters. And again, we still think the environmental concerns are still not entirely clear. These things must be addressed. Consumer acceptance of nanotechnology is a big problem out there right now, but these are the obstacles we have in front of us. But I think the future looks extremely bright for using nanotechnology. It's no silver bullet, as we say, but it's certainly going to be a new tool for us to be able to give us a little bit more yield in maximizing the productivity in some of these fields. I want to thank all the people that have worked with me. My technician, Peter Thiel, he just retired, but he was a crucial member in helping me accomplish all that. All of my colleagues here at the Connecticut Ag Experiment Station, Dr. Gardia is at University of Texas, and Matthews Perret is at the South Florida, Florida State, University of Florida, excuse me. And also I'm a member of the nanotechnology, the Center for Sustainable Nanotechnology, and it's a National Science Foundation sponsored grant here. And basically, I and one other person are the only plant people in this group. These are all chemists, but so I feel really fortunate to be able to work with such smart kids that are creating these amazing products for us to be able to test on. So with that, I think I'm pretty close to being done, and I'll be glad to answer any questions.
Yes. So, Dr. Elmer, so it's very nice talk we have heard just now, really based on your practical experience. And one good thing I have seen here, so whatever experiment you have conducted, so you have repeated the same experiment for 3 seasons, 4 seasons. This is a long time experiment. This is very convincing. That's why that all the results. So another one, I have understood this nanoparticles, copper nanoparticles, especially copper oxides. So you have applied as a micronutrient, not as a fungicide. So here, actually, I am also working in this line, but we are using these nanoparticles as fungicides, but you are indirectly using as a micronutrient so that it is boosting the health of the plant, seedling, and finally, so they are able to challenge the invading pathogen. But my question is there, here, when you are handling these nanoparticles, so you are allowed to use in open field or in a confined area in a greenhouse or glass house? That is actually our main problem. Yeah, is not allowed to use in open field.
Right. The first grant we wrote back many years ago was not funded because we did not claim to have a good safety procedure for applying these in the field. We checked with the United States Department of Agriculture. There were no guidelines for using nanoparticles in the field, but we decided, well, let's just not even go there. Let's just stay with treating these things in a greenhouse, in a glass house, in a very confined area, and and we make sure it's very safe, and we apply them, we allow them to dry, then the next day when they're dry, we take them and put them into the field. So far, we can apply these in the field, but right now we're trying to not do that because that is we're just trying to reduce the exposure of these nanoparticles in the field. But your point is, my colleague down in Florida, Dr. Perret, he is actually putting nanoparticles into backpack sprayers and going out and spraying tomato plants. So using these nanoparticles as a bactericide, and he's able to reduce the amount of copper significantly by using the nanoparticle form versus the copper hydroxide fungicides that would normally be used. But in that situation, again, there's no guidelines in the United States for using nanoparticles in the field, which is, I think, something we need to work on.
Thank you. So another one, so you are synthesizing nanoparticles yourself, or you are using some commercial formulations?
I'm sorry, I didn't understand.
So you are synthesizing nanoparticles yourself, or you are using the commercially available formulations?
Both. Originally, we were using the commercial preps that we were getting from a company called US Nanotechnology Research, I believe. And we were just getting the oxides, the manganese, the copper, and the zinc oxides from this company. Since we started working with the Center for Sustainable Nanotechnology, we've had access to a lot of students who have been synthesizing their own nanoparticles. And these products are superior to the commercial ones, but they are probably not available to the grower yet. But we've been using them in many different forms of copper phosphates. They've been able to create the nano sheets. They are loading these things onto carbon dots. And right now, this morning, I learned about a product called mine scenes. And they're two-dimensional. They're just basically, they're like 100 nanometers wide and 1 molecule thick, so extremely thin. And these things have a very, they seem to be very effective for delivering nanoparticles. So that's a new way. So right now, all the synthetic stuff that we're using is coming from these laboratories. It's not commercially available.
My last question is, so regarding application parts, most of your application, this experiment you have done by applying those copper nanoparticles foliar application. And since in case of vegetables and also flowering plants, these seedlings are transplanted. So whether this root deep treatment will be more effective than foliar application?
That's a good question, and we've yet to do that. We're starting to look at infusing these products into seeds and planting them out and seeing how that would look. Again, if you had an opportunity to dip the roots before planting, that would be fine. Again, that's going to be putting more of the nanoparticle in exposure into the soil. And that's a concern. We want to limit all exposure of the nanoparticle in the soil. But if you could apply them and then maybe allow the plants to drain as well, you know, that might be a better way, because you could be putting it directly onto the affected tissue. One reason I was looking at foliar application is that many times I work with perennial plants and we don't have access to the root systems. And we don't see the disease until the 2nd or 3rd year, like in strawberries or asparagus. These are perennial systems. So that was my first introduction into using nanotechnology, as I was trying to see if I could use them as a rescue treatment. So that's why I went with the foliage route there. But that's an excellent idea. I think you should do it.
Yeah, thank you. Thank you. Yeah, any others from audience?
Good morning, sir. So
Morning.
I have a question. Yeah. So yes, first of all, sir, I would like to say thank you, since many of my questions has already been answered. But my mind is still linger on one question, sir. So I have been wondering, sir, how exactly these nanoparticles affects the pathogens. As we see in other fungicides like carbendazim, it affects the biosynthesis of DNA, and for example, in case of mancozeb, it affects the enzyme having the sulfonyl groups. So likewise, sir, I was wondering, that how this copper nanoparticle exactly affects the pathogens, sir, if you could answer this.
I don't believe that the nanoparticle is in concentration high enough to have a direct effect upon the pathogen in the soil. We're applying to the foliage, in these young plants, and then we're probably not beginning to get infection until maybe 10 days later when they're in the soil. What I think the effect of the nanoparticle is on the host by increasing these defense mechanisms, so that when fusarium or verticillium or whatever, when it invades that root, there's usually a reaction of the plant, and at least in asparagus, it's very clear it lays down a lignin barrier very quickly, and the speed in which it can lay down that barrier can frequently mean resistance or susceptibility. And I think that the speed of that laying down that can be a function of how much copper is available to that cell.
Okay.
So I think that's the main mechanism that we're seeing with the plants. But if you, as you know, as Dr. Robin just mentioned, if you were to expose the roots to these things, it could be possible there'd be enough nanoparticles to directly affect the pathogen. Because copper is very toxic. I'm not sure if it's a DNA effect. I can't remember exactly what the mechanism of copper is against microbial pathogens, but it is an antimicrobial.
Okay. Thank you, sir. Thank you.
So any more questions from other audience? Very good number of participants I have seen.
I think there are some questions in the chat box. Some important questions.
there are.
Dr. Lakshman Prasad has asked, what is the best organism for green synthesis of nanoparticles? I think one of the participants has asked this question. If Dr. Wade can reply to this, yeah.
That's a good question, and I don't have a good feel for that yet. I'm in fact, in a few weeks, I'm gonna be getting a new scientist visiting us from Tunisia, a young woman who's going to be looking at green synthesis. Right now, you can make, as I understand, you can make these nanoparticles from just about any type of organism, as long as they have enough of the extract and the reducing agents in their cell systems to cause the nanoparticles to form. But you can use plants, but as I understand, with plants, you have a chlorophyll, and then you have you have to kind of clean it up a little bit more. Right now, we've been using some bacterial systems in the laboratory. And I don't know what the bacteria are, but you go into the literature. I have to admit, Americans are way behind in using green synthesis for nanotechnology. That's because we've had access to these laboratories where they've been making these interesting things there. I have to admit, I'm not as knowledgeable about that. The next question, what's the cost benefit ratio and yield after using nanoparticles in the field crops? As I mentioned, where we've worked it out, it's been tremendous when you think about the cost of these tiny little nanoparticles. We're putting on 1 or 2 milligram per plant, and that's coming into just pennies per plant. And then we're getting sometimes 30 to 40% more fruit off of that plant. So it could be a sizable benefit in yield in that way. What's the characterization copper nanoparticles prepared from Ossicum sanctum leaf extracts? That's a basal type. I've actually never worked with preparing those. I really wouldn't know. But that would be something. I'm sure there's some people out there that have actually used the extracts. If you went and you looked in the literature, I'm sure you'd find many articles that discuss that. So I really am not very knowledgeable about the green synthesis. What is the effect of capping agents on nanoparticles? You mean like coating and so forth? That's very interesting. We found that when you coat things like, nanoparticles of sulfur by themselves are very effective in causing suppression of various diseases. But we have a student now, a postdoc, and she finds that when you coat those nanoparticles with stearic acid, you get about a 20% increase in yield in the field. you get about 20%, you get about anywhere from 10 to 15% more disease suppression. And we don't really understand why that is. It may have to do with just dispersion. Nanoparticles, one thing about nanoparticles, as soon as you put them in the water, they start to become microparticles. So they start to coagulate. So keeping these things dispersed is a major problem in nanotechnology. And I think coating them might be allowing these things to stay dispersed better. So that's one of the main things that we've been looking at, the coating. You can go online now and look at some of these companies, and they are providing nanoparticles that are coated, but we've been trying to do our own. But stearic acid, and I don't know enough about the chemistry to know why stearic acid is preferable to citric acid or anything else, but we have found that that's an amazing coating mechanism. I don't see any other questions in there.
Any other student, also faculty? So I think no more question. I think we have come. Yes.
There's one more question. Can you comment on the toxicity of nanoparticles? Very good question. We kept thinking that we were going to see toxicity. I've gone up to 2 grams of 2,000 parts per million of these things on eggplants and so forth. I've seen no damage in the greenhouse. So far I've seen nothing that I can call toxicity. Now, if I were to use the salt, I've used a copper sulfate that rate, I would burn the leaves. And so it does seem to be, the nanoparticles seem to be very safe in terms of plant damage. I've always rated these plants for phytotoxicity after I put them on there, and I've never seen any.
I think no more questions. My last question, and I want to wind up this program. It's Dr. Elmer, so that copper oxide nanoparticles you are using, what is the particle size, range of the particle size?
That's a good point. Yeah, the nanoparticles that I've been using of copper, and then the 3 studies I showed you, they come from a company, and they're about 30 nanometers.
30.
So that's about 30 nanometers, right?
Okay.
And if that's like the size of a soccer ball to the earth. That's the range there. So it's a very small molecule.
Right.
And there's one last company. What happens if excess toxicity affects consumers? That's also a good question. Now we're putting these things on at very, very young plants, and then we're harvesting fruit months later. So if there is any movement of these nanoparticles into edible tissue, it's so diluted that I don't think we're going to have a real problem with that yet. If you're putting on very high rates on something like lettuce that you're going to eat in a few weeks, there's going to be a problem. So, but in the systems I'm working on, we don't have reason to think that there's any toxicity to the edible portions.
Thank you. So before winding up today's lecture, so is there any other question? So I request our president of Indian Phytopathology Society and the president-elect, both are connected. So first I request Dr. Pratibha Sharma, president, to give her comment. And next to her, so I request Dr. Rakesh Pandey.
Yes, I'm most welcome, Dr. Ware. I can understand in the midnight you are delivering this lecture, and it's a great opportunity for all our Indians to listen to this lecture, and the pains which you have taken, and you were very cooperative and enthusiastic about this lecture. And I think the whole lecture went off it was very practical actually, because when we are talking about nanoparticles, we are all very concerned about that. Are they really making some effect in the yield, or whether they are really applicable in the field. So I know my all Indian young participants and scientists, what they were wondering was whether it will really help us in increasing the yield, or does it really help us in managing the pathogens. So all the questions in your slides were very well answered, and we could really understand. And actually it was very good also because you have worked on flowers, you have worked on vegetable crops. So as such, it was a good range of crops have been covered by nanoparticles. And that too, these nanoparticles actually we are linking with the more with the yield, but sometimes it is very good to listen that yes, some of the these nanoparticles which may be belonging to copper or zinc or some these elements, they are helping us in some way managing the diseases also. So I think it can be a good combination when we are talking about nowadays, when we are talking about the nutrient management. So actually, it was a very good reflection on the nutrient management through nanoparticles. And I think Dr. Govey himself works on nanoparticles, so he was very much interested. And all other participants who have asked these questions have found it very educative, and hope so that it really it is how will it go in at commercial level. Commercialization actually, when it goes to the market, it comes to the farmers, will it be very effective? Because there is another view also when we are taking that because we have this question on this nanoparticle section. But nevertheless, the work we found it to be very educative, very, and hope our all participants will be really benefited, and they have understood it very nicely, because very simple, very simplified. You have really gave the explanations. Thank you, Dr. Wade, for all your this presentation, and really your great enthusiasm to be the participant in the midnight also. Thank you very much, Doctor. Thank you.
Thank you, madam. So I would request Dr. Rakesh Pandey, our president-elect.
Yeah. First of all, I congratulate Dr. Wade and his team for doing a wonderful work. And really, we are all Indians, we are benefited from his lectures. We are using nanoparticle for the biomedical sciences, but in plant sciences, it's a very good result, and hope these different size of the nanoparticle will have impact on the crop yield. It's a wonderful work. And again, I congratulate Dr. Wade. It's a great thing, and hope this nanoparticle will also increase the yield of the plant secondary metabolites, because I am the person who is working on plant secondary metabolites. Thank you very much, Dr. Wade. Congrats again.
Yeah.
Thank you very much.
Thank you.
Okay. I think Dr. Suresh, now this is my part to give vote of thanks. Dr. Suresh Gawande.
Yes, sir, I request Dr. Govey to deliver vote of thanks.
Okay, thank you. So really we have learned so many information and date to practical results from today's lecture. And despite of this odd timing and also busy schedule, Dr. Wade Elmer is able to help us contribute to our society, Indian Phytopathological Society. So that's why on behalf of our society, profusely I'm thanking you. And also all the audience, they are joining from different parts of our country and also from abroad. I have seen Dr. Dilip Lakshman from US, he's also connected. He also enjoyed your lecture. So I thank all the participants and attendee, and also some of the viewers, they are connecting through our YouTube and also through our society website. So on behalf of society, I am thanking all of them. And I hope so all people will be joining in our next lecture series also. And I once again thanking Dr. Wade Elmer, and I hope you will be cooperating in future also. Thank you.
Thank you.